Lecture sarcoplasmic reticulum and sarcoplasmic mitochondria discussed કરી, right? Now, today the structure of contractile protein. બાકી બધું તમે પછી રીડ કરી લેજો. Structure of contractile protein. Which are the contractile proteins? Actin and myosin. બરાબર ને? So actin, troponin and tropomyosin, how they are arranged. So now first we are talking about the thin filaments. Okay? Structure of thin filament. Understand this. First of all, this is known as G-actin. G is nothing but globular actin. Okay? Globular means one molecule of it. So with the help of, this is very, very important, with the help of Mg+2, okay, with the help of Mg+2, this globular actin is spirally arranged. And form the F-actin. So this is known as F-actin, that is known as filamentous actin. At the time of the formation of the muscle, generally in our body, I am talking about in the embryonic state. First of all, G-actin was there, and then G-actin is properly arranged spirally, okay, and form the filament-like structure, and that is known as F-actin. Now, here I will change all the things. Now, as you know that according to the video, these all are red color molecules. Actin molecules, if you have remembered that, fine. So now I will make the dots on each and every molecule. So what do you give the name for this dot? What do you give the name for this dot? Which is covered by purple line. So just over that, if you remember, this red dot is known as myosin binding site on actin molecule. Red dot is known as myosin binding site on acting molecule. Okay. And now this black colored filament, that is known as, yes, tropomyosin. So this is known as actin molecule. Fine. Actin molecule, myosin binding site on the actin molecule, and tropomyosin. Up to this, everybody clear? And now, at particular interval on the tropomyosin, 3 molecules are there. Okay, so these molecules are known as, yes, troponin. Troponin can be represented in the short form TP. Troponin, TP. So here, in the troponin 3 molecules, fine, troponin I, troponin T, and troponin C there. Okay, TPI, TPT, and TPC. 3 troponin molecules are there. Understand, everybody? કાંઈ નહીં થોડું ખાલી એક જ વીક માટે સસ્પેન્ડ કરી દઈશ. No punishment, no not down in the progress book. Return. Completed. this is known as thin filament, and here the G actin is converted into F actin. But important MCQ with the help of Mg plus 2. Okay, and another MCQ is very important for NEET exam, that during muscle contraction, which two ions are very essential? So answer is magnesium and calcium. These two ions are very important at the time of contraction of the muscle, because you know that G actin is converted into F actin with the help of Mg plus 2, and during muscle contraction calcium ions are very helpful. Fine. So just over the actin filament, actin molecule, fine, tropomyosin is totally covered, the myosin binding site on the actin molecule at the time of relaxation, and just over the tropomyosin filament at particular interval, three molecules of the troponin are there. So these three molecules of the troponin are troponin I, troponin T and troponin C. The I, T and C, find I, T and C, the name is given according to that. Troponin I majority generally inhibit the muscle contraction. At the time of relaxation, the troponin I can inhibit the muscle contraction process. Troponin T maintains the attachment and the structure of the tropomyosin. And the most important thing is this: out of all the troponin molecules, troponin C is very useful for attachment of calcium ion during muscle contraction. If you have remembered that video, the calcium ion move and attach to the troponin molecule, and at that time tropomyosin filament is contracted and myosin binding site on the actin filament is open. At that time ATP is broken down on the myosin filament, and with the help of that with the help of that energy, the cross bridge formation occurs. Okay, so that calcium is attached to the troponin C. Please remember this. And now the new thing for you. We have easily told that calcium ions are released from the sarcoplasm. But actually in the sarcoplasm, I mean to say the cytoplasm in the muscle. So sarcoplasm in the T system is present. T system is also known as transverse system, which stores the calcium ions. So when our nervous system gives the impulse to the muscle for the contraction, at that time, as you know, that the nervous impulse moves from the brain and it is transmitted through the neuromuscular junction. Then that impulse is now entered in the sarcoplasm. Impulse is now transmitted to the sarcoplasm, and when the impulse is there in the sarcoplasm, at that time sarcoplasmic mitochondria release the energy, and simultaneously the T system which is present here in the sarcoplasm release the calcium ions. Calcium ion move towards the troponin molecule and attach at troponin C. At the same time ATP is bind on the myosin filament. And then after contraction, the myosin head is moved towards the thin filament and cross bridge formation occur for pulling the thin filaments towards the H bed, M line. In this way muscle contraction occurs. Fine. So this is the structure of the thin filament only. Thick filament is remaining now. Actin and myosin binding formation occurs without ATP, and cross bridge breakdown in the presence of ATP. So these two are very important points. Okay, anybody has any doubt here in the thin filament structure? Then I will continue with the thick filament. Any doubt? Okay, fine. The thick filament, very short point. Fine. So the thin filament is made up of actin, troponin and tropomyosin. The structure is over. Now the thick filament Maybe. See, thick filament, it is made up of only myosin. Fine, but the unit of the myosin filament. This is myosin filament. Similarly, you have remembered that actin filament is there. So when actin filament is made up of the actin globulins. Similarly, the myosin filament, it is made up of meromyosin. So the unit of the myosin filament is meromyosin, and here the two types of meromyosin there, heavy meromyosin and light meromyosin. But here the myosin filament both are spirally arranged together, and the site at which crossing arm formation occurs, that is known as neck region. Because I think you are confused between this neck and that neck. Okay, let me clear that. So here, this is the myosin filament. Myosin filament. And this is the neck region from where crossing arm is originated. And in the crossing arm, again 3 parts are there. Okay, so in the crossing arm, the topmost part is known as head, then middle part known as neck, and the terminal part which is known as tail. So this tail which is arranged to the neck of the myosin filament, and in this way thick filament is generated. Is that clear about this two neck word? Fine. Now, the two things are very important here. In the head region only, okay, in the head region only, 2 sites are there. This is very important. At the time of muscle contraction, it is very essential. Fine. So, on head, 2 sites are there. One is known as ATP binding site. So, previously I have told to you that from the sarcoplasmic mitochondria, ATPs are released. These are attached here. In the video, if you remember that ATP is joined to the crossing arm, that is known as ATP binding site. Okay? And on the top of the crossing arm, one dot is also there. That is known as actin binding site on the myosin filament. And previously, the red dot is myosin binding site on the actin filament or actin molecule. They alag છે. They alag છે. બે સરખું થઈ ગયું. Red dot is myosin binding site on the actin molecule, and here actin binding site on the myosin filament. So generally what happens, that calcium is bind to the troponin C, that tropomyosin is contracted. So on the actin filament, myosin binding site is open. Okay? Then ATP is bind to the ATP binding site here. It is broken down, and with the help of the energy, that cross arm move upward or move in particular direction to bind with the actin molecule to pull the thin filaments. Okay? So crossing arm consists head, neck and tail. So for easily understanding purpose, you remember that head is mad up of head. H to H. Okay, so head is made up of heavy medomyosin, and remaining neck and tail are made up of light medomyosin. Okay, that's why I will make a round here, H to H. Head is made up of heavy. Clear? So this is about the structure of the contractile protein, and mechanism of contraction is also over. Fine, you read it from the textbook. Then in the next lecture, here I can stop now, but in the next lecture, I will continue with the fatigue muscle, one thing else, and types of muscles, red muscles and white muscles, and then we will continue with the skeletal system. Clear? Okay, so here I can stop now. Fine.